Welcome everyone to today's Explainer. We are jumping right into the incredibly high-stakes world of clinical pharmacology today, and we're looking at the relentless scientific search for the absolute perfect medication dose. I mean, it sounds simple enough, right? You get sick, a doctor prescribes a pill, you take it, and boom, you get better. But behind the scenes of that seemingly simple transaction is a massive, complex biological balancing act.
So think about this for a second. Why can the exact same dose of medicine completely cure one person but cause serious harm to another? It's a pretty fascinating paradox, isn't it? You could literally take two people with the exact same diagnosis, give them the exact same amount of a drug, and see radically different outcomes. And this whole baseline problem, it's rooted in our human biological variants. We're talking about things like your age, your weight, your organ function, and especially your unique genetics. Our bodies just don't process chemicals identically. And when it comes to medicine, when the stakes are incredibly high, guessing just isn't good enough.
Okay, let's dive into this. The solution to all that biological variance is something called Therapeutic Drug Monitoring, or TDM. Basically, TDM is the clinical practice of measuring specific drug concentrations right there in a patient's blood. The ultimate goal is to optimize that medication therapy. It's all about a very delicate balance. On one hand, we absolutely must ensure efficacy, meaning the drug is actually working and doing its job. But on the other hand, we have to simultaneously minimize toxicity, making sure the drug isn't building up to dangerous levels in your system.
Section 1. The Dosage Dilemma.
Whenever you swallow a pill or get an IV, two distinct scientific processes kick off at the exact same time. First up is pharmacokinetics. The absolute easiest way to remember this is it's what your body does to the drug. How fast do you absorb it? How does it distribute through your tissues? How does your liver metabolize it? And how quickly do you eliminate it? Then on the flip side, you have pharmacodynamics. This is what the drug does to your body, specifically the actual effect it has when it hits its target receptor sites. Understanding both of these principles side by side makes it crystal clear why dosing is never, ever a one-size-fits-all scenario. I mean, if your metabolism is faster than mine, your pharmacokinetics change entirely, which means the drug's pharmacodynamics inside you will be totally different too.
And that brings us to the concept of the Goldilocks zone. In pharmacology, we call this the therapeutic window. If the drug concentration in your blood is too low, well, you have poor efficacy. The medicine basically isn't doing anything. But if the concentration is too high, you hit severe toxicity and start experiencing dangerous side effects. TDM is the precise scientific tool that clinicians use to find that concentration that is, just like Goldilocks said, just right.
Now, obviously, we don't need to monitor every single drug this closely. So when is TDM actually indicated? Well, there are four main scenarios. First, for drugs with a narrow therapeutic index, which just means the gap between a helpful dose and a toxic dose is incredibly small. Second, when there's marked pharmacokinetic variability, meaning patients process the drug super unpredictably. Third, for drugs with severe dangerous side effects. And fourth, for drugs where it's just really hard to tell clinically if they are working until it's far too late. To put that into perspective with some concrete examples, doctors rely on TDM for anti-epileptics like phenytoin and valproate, heavy-duty antibiotics like vancomycin, and immunosuppressants given to transplant patients like cyclosporin and tacrolimus. A few other common ones are digoxin, lithium, and theophylline.
Section 2. The TDM cycle in practice.
Out in the real world, in a hospital setting, TDM is a continuous cyclical process. It's like a giant feedback loop. It starts with a medical judgment. A doctor diagnoses you and prescribes a drug. The drug is administered, and then a blood sample is collected. That sample is rushed to the lab for analysis, where they use advanced machinery to figure out the exact concentration of the drug in your blood. Finally, the clinician interprets those results and makes a highly personalized adjustment to your dosage. And then, the cycle begins all over again.
Breaking that cycle down into distinct steps, there is one absolutely crucial detail we literally cannot overlook: timing. Step 1 is administration, sure, but Step 2, sample collection, has to happen at exactly the right moment. Clinicians are usually targeting either trough levels, which is the absolute lowest concentration of the drug in the blood right before the next dose is given, or peak levels, which is the highest concentration shortly after a dose. If you draw the blood at the wrong time, well, the laboratory analysis in Step 3 is going to give you totally misleading results, which means the interpretation and dosage adjustment in Step 4 could actually harm the patient. Timing is quite literally everything. During that laboratory analysis phase, scientists rely on some seriously specialized techniques. The two main categories here are immunoassays, which use antibodies to detect the drug. Think of methods like ELISA or EMIT. And then you have chromatographic methods, which physically separate the blood's components, like HPLC. Choosing between these techniques really just depends on the specific drug being tested, the resources the hospital actually has on hand, and the exact level of accuracy required for that specific patient.
But here's the catch. Once the lab spits out a number, the clinician's job is far from over. They have to interpret that number through a really complex human lens. They have to account for a patient's age, their weight, and their unique genetic variations. They have to look at organ function, specifically how well the liver and kidneys are working to metabolize and clear the drug. They also have to consider any other medications the patient is taking that might cause a nasty drug interaction. And finally, they have to factor in adherence to therapy. Basically, did the patient actually take the pill when they were supposed to?
Section 3. High-stakes case studies. So why is this all so important? The stakes become incredibly clear when we look at specific drugs. Let's take phenytoin, an anti-epileptic drug used to control seizures. It has a strict, incredibly narrow target range of 10 to 20 micrograms per milliliter. Because it has nonlinear kinetics, meaning it absorbs and builds up really unpredictably, even a tiny increase in the dose can cause blood levels to skyrocket, leading to severe toxicity. But dropping below 10 means the patient might suffer a life-threatening seizure. Then we have vancomycin, a heavy-duty antibiotic used for severe MRSA infections. Here, clinicians meticulously monitor those trough levels we talked about earlier. Why? Because if the dose is too high, it causes nephrotoxicity, severe kidney damage, and ototoxicity, which can literally cause permanent deafness. TDM is often the only thing standing between curing the infection and causing permanent harm.
Okay, take a breath, because I know that sounds pretty intense. I want to reassure you for a second by sharing a direct quote from the source material. Majority of medications do not require TDM. If you're just taking a standard over-the-counter ibuprofen for a headache or everyday blood pressure meds, you absolutely do not need your blood drawn every day. This intensive TDM process is reserved specifically for those unique, high-danger, narrow therapeutic index drugs we just went over.
Section 4. Global Realities and the Future.
But we have to talk about the logistical reality of TDM because it's pretty stark. This isn't just a doctor making an educated guess. This requires continuous, coordinated communication between doctors, nurses, and laboratory staff. Furthermore, those chromatographic machines and specialized analyzers, they are highly advanced and extremely expensive. And frankly, this logistical reality makes TDM very difficult to implement universally.
To see how this actually plays out globally, the source material specifically examines the challenges of practicing TDM in Nigeria. In many resource-limited settings, there's severely limited access to the advanced laboratory methods required for accurate testing. Cost acts as a major, sometimes totally insurmountable barrier for patients and clinics alike. There's an urgent, ongoing need for capacity building and better medical infrastructure. However, despite these very real hurdles, the research notes that introducing TDM in these regions holds massive, completely untapped potential for vastly improving patient outcomes, especially for people managing complex chronic diseases.
But all of this builds into a seriously exciting future. The science of therapeutic drug monitoring isn't just standing still. We are looking at a future where TDM is integrated directly with pharmacogenomics, which means we'll use your specific genetic code to predict exactly how you'll process a drug before you even take a single pill. We're moving towards point-of-care testing, where a doctor might be able to check your drug levels instantly, right there in the clinic, rather than shipping blood off to a distant lab. We are even seeing artificial intelligence stepping in to help clinicians predict the perfect, individualized dose. And all of these advancements, they aim to drive the expansion of TDM right into those resource-limited settings, making safe, personalized drug therapy accessible to everyone.
Which brings us to our final thought for this explainer. Look at the horizon of clinical pharmacology. With artificial intelligence and pharmacogenomics advancing as fast as they are we entering an era where the one-size-fits-all pill is entirely a thing of the past? Are we approaching a day where every single prescription you ever receive is uniquely and perfectly tailored to your individual DNA? It's an absolutely fascinating question, and the science of TDM proves we are getting closer to that reality every single day. Thank you so much for joining me to decode the science of the perfect dose. Keep asking questions, keep learning, and I'll catch you in the next explainer.